Yeah.
Because my mouse won't go there. Okay, thank you. Okay, so sorry, but I don't know why my mouse isn't working, but it's not working. Maybe that was my problem before, my mouse wasn't working. So anyway, so we're going to be talking about gene regulation, and so this is just an interesting thing that was found through studying fruit flies, is that gene expression, proper gene expression, having the right genes on in the right parts of the developing embryo of a fruit fly greatly influence the structure that comes about when the fruit fly is an adult. And this is an interesting transition because remember, a fruit fly is an insect that goes through that larval phase, so they're an embryo and then they go through a larvae before they become the adult. But you can see here this middle gray section right here, this represents the chromosome, and these are genes along the chromosome, and these genes are expressed in almost the same order that they're found on the chromosome along the length of the embryo, and then those same gene expressions influence the structures of the adult fruit fly. And if you don't express these genes during development, these structures don't end up coming. So if they're not expressed during the embryo here, you end up with a fly without the adult structures here. So it's very important to establish some gene expression pathways very, very early in development. And the other thing that's interesting is if you misexpress, if you take a part that's supposed to be expressed here, and we're going to do that in a second, we're going to take this gene right here, it's called antennapedia, normally is expressed in this segment that gives rise to this second leg and the wing part of the fruit fly. If you misexpress that in this segment, so if you put it under the wrong control, if you express it in the wrong place at the wrong time, what happens is the fruit fly ends up having, instead of antenna coming out of its head, it will actually induce the growth of legs coming out of the head, as if the fruit fly were to think that was its middle segment where the legs were supposed to be. And you can see here, this fly got to adulthood, right? There's eyes, there's the head. And so this is what happens. And this is one of the reasons why fruit flies are so amazingly awesome to study, because in a animal other than a bug, in like a mouse as a model system, for example, these individuals would likely never be born and make it to even a baby phase as opposed to adult. But in fruit flies, for whatever reason, many of these huge changes can make it to the adulthood. So here's another example. So if I go back to that original picture, right here in this segment here, we express a gene during embryogenesis called ultrabithorax. And it makes this green segment have a leg, and it also makes that green segment have this thing called a haltere. All right, so they're really just for balance on the fruit fly. So if we misexpress that same antennapedia in this segment, instead of ultrabithorax, what happens is the fruit fly just doesn't have this segment with the haltere, and instead makes a whole repeat of that second segment, so that the fruit fly actually has kind of two backs, it looks like there, two thorax, and 4 wings instead of 2 wings. So pretty cool. And this is just gene manipulation. All we're doing is expressing a gene during embryo development in a different place than it would normally be expressed. So this is why it's so important to understand how gene expression is controlled. All right. So here's just another one. So this is misexpression of the genes that give rise to the eye in other places. And you can see this eye is growing on the leg of the fruit fly. And you have another little eye over here. And this is turning off the eye gene altogether. So no eye forms on this fruit fly at all. And so fruit flies are just so amazing for this purpose, because you can really just really manipulate the genetics and see real outcomes and really start to understand how that gene expression, how important it is. So it's very well studied in fruit flies. And it turns out that a lot of these early embryonic gene expression patterns in fruit flies are also the very embryonic gene expression patterns in all animals. Some of the early ones just dictate where's the head going to be versus where's the tail going to be? Where's the right going to be in the left, and the dorsal and the ventral, right? And so those, we can study those, and the expression pattern in a human embryo is really no different. Okay, so we're going to start off talking a little bit about gene expression in E. coli today, and then we're going to do a review of how gene expression is controlled kind of all over the place. So keep in mind that in bacteria, there's no separation of transcription and translation by the nuclear membrane. And so they can happen simultaneously. Also keep in mind that the DNA in a bacteria cell is smaller. There's less of it. They still have a lot of genes, not nearly as many as we do, but they still have a lot. What they lack is they lack a lot of extra, what we call like regulatory DNA. They have, instead they just have, most of their DNA has purpose, has gene encoding information on it. Okay, so I think we don't need to do that one. Okay, so let's skip ahead to this one. Well, let me go back to this slide. So keep in mind that in every cell, you only want to express the genes, and remember what that means. That means make the proteins go through the whole central dogma. You only want to express the genes that are necessary and needed for that particular cell to function. It costs energy, it costs time, it costs resources to making those proteins. So you only want to make the ones that are necessary. So for example, these two cells came from the same individual. They have exactly the same DNA in them. However, in this cell that's going to become a neuron, we have to express neuron-specific genes to make proteins that make it a neuron. In this one that's going to become a skin cell, we're going to only express cells that are necessary for skin cells, but we're not going to express the neuron-specific genes. All right, so keep in mind that there's this, it's called differential gene expression, meaning that we have different genes turned on in different cell types at different times to make sure the cells follow the path that they're supposed to follow. And we regulate that gene expression in a whole variety of different ways that we're going to be talking about. So first, we're going to talk about it in prokaryotes. All right. So one of the things that's interesting for prokaryotic genes is that not every gene has its own promoter. So what we just saw in the last chapter was we saw like the structure of a gene. We saw it had a promoter, the coding sequence, a terminator. We made the RNA for that one gene. In bacteria, the genes are located on the chromosome in such a way that they are end-to-end with each other, and they code for proteins that are going to participate together in a pathway somewhere else. So if you look here, these are 5 genes, 1, 2, 3, 4, 5. They're right next to each other. They're under the control of one promoter. They end up making these 5 different proteins, and I'm just going to skip ahead a slide. And those 5 different proteins function together. Hold on, I'm coming. They function together to get the job done. All right, so here are those 5 genes. In order to get the last product that we need, we have to express this enzyme, 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 we have to express this enzyme in order to get this whole process to happen. So what we do is, because they're in the same pathway, we are going to put them together on the chromosome as well, and then just express them all at the same time under the control of one promoter. All right, and that is called an operon. Sorry, I'm going to shut my door. It's not my kids today, it's my husband that keeps being noisy. Okay, so an operon is a set of genes that are under the control of one promoter, and they make proteins that function together in a process or a metabolic pathway or something like that to get the job done. All right, so make sure you know the definition of an operon. So again, here are those 5 enzymes that we have to make in order to get there. All right. So, like I said, they're functionally related genes that make functionally related proteins, all right, and they're under the control of one promoter. All right, now let's look at the structure of the operon. It's a little bit different than the structure of a gene in a eukaryote, all right? Like I said, there's only one promoter. Instead of having a promoter before each of these genes, there's just one. So we're only going to do one set of transcription that goes the whole way through X, Y, and Z. The promoter is the same function as the promoter in a eukaryote. This is where the RNA polymerase is going to start and start the whole bind and start the whole transcription process. And then we have two other DNA sequences that are important for keeping control of this. We have a DNA sequence called an operator, and an operator is a sequence to which a protein called a repressor can bind. All right. So it's like the olden days telephones, like I'm talking like before me even, like when my mom was little. It would be that she would just go pick up the phone off the wall, and there would be a woman, an operator, and my mom would say, hey, I want to call my cousin, and the operator would say, okay, hold on, and then she had this big switchboard in front of her, and she would basically switch a couple switches to connect the line from my mom's house to the line from her cousin's house, and now they would be able to talk to each other, right? When the conversation was over, the operator would be able to disconnect those lines, and they would no longer be able to talk to each other. So this sequence is kind of it's called an operator because it acts just like the old day telephone operators, because it's this sequence that can make the difference between whether we're going to have gene expression and transcription of this operon or not, okay? And it is under the control of whether or not a protein is bound here or not. Okay, now the protein that binds here is called a repressor, and it's got its own gene with its own promoter, and we're just going to call that gene I for right now. All right, so the operator is a DNA sequence that acts as an on-off switch. A protein called a repressor protein binds to the operator. If a repressor is bound to the operator, that turns the gene expression off. All right, so when the operator is on, that means the repressor is not bound to the operator. All right, we're going to see some examples. And so basically, if you think about it, if something were sitting right here on this spot, if the repressor protein's sitting here, right here on this spot, then the polymerase that's sitting on the promoter is basically going to have a block on the road that it can't get through. You take the repressor away, and the polymerase can then travel straight through. All right? And just keep in mind, the repressor protein is its own protein. It's under the control of a different gene and a different promoter. One of the things we're going to find is that the repressor can be in an active form, which means it binds to the operator, or an inactive form, which means it doesn't bind to the operator. And there are things that change it from one form to the other. Okay, so now let's look at some examples. All right, we know, let's think about E. coli in our intestines, right? The E. coli in our intestines have to adjust their metabolism to be dependent upon whatever we eat. So if we eat, for example, if we eat a sweet roll, then the E. coli is going to be bathed in sugars and fats. And so the enzymes necessary to break down sugars and fats are going to be needed in that cell. So the cell is going to be very busy making those enzymes so that we can get benefit from those nutrients. However, if we drink a glass of milk, that changes the E. coli's environment. Now, instead of being glucose and sucrose sugars, now the E. coli is surrounded by a lactose, which is a different sugar. And so the bacteria now has to start making the enzymes involved in breaking down lactose. All right? So that means the genes that are necessary for breaking down lactose, which involves enzymes called lactase, have to be turned on so that we can break down the lactose. However, someone like me who's lactose intolerant but doesn't drink milk, I don't ever need to turn that operon, or my bacteria in my gut never need to turn that operon on. So that is, we would have it in the off position for most of the time. Okay, so let's think about what this is. There are 3 enzymes necessary to break down lactose. All right, we'll call them LAC-Z, LAC-Y, and LAC-A. They're grouped together on the DNA. The genes are grouped together on the DNA under the control of one promoter. So we call this the LAC operon, the lactose operon. The operon has those 3 genes and 2 control sequences, the promoter and the operator. All right, so let's think about the first scenario. Let's think about me. If I don't ever drink milk, do I want the repressor to be bound to the operator or not? So there's no lactose in my system. Do I want the repressor to be bound to the operator or not?
No.
We do want it to be bound. That's exactly right. We want it to be bound because we don't want those genes to ever have to get transcribed because I haven't eaten any lactose. Okay? So in the absence of lactose, the repressor protein will bind to the operator, and that's basically like a block in the road for the repressor to get through. I'm sorry, not the repressor, the RNA polymerase to get through. So we have transcription turned off. However, let's just say I drink a glass of milk, right? Well, now I want to make sure these genes get expressed, so I've got to remove this repressor from the operator. And so in that case, let's see if I have a picture here, we are going to inactivate the repressor so that the polymerase can read through and make the whole RNA. All right? The way that happens is actually from the sugar itself in this case. So in a normal situation where I don't have any lactose around, the repressor protein is made and is capable of binding the operator. It blocks the polymerase from going through. I drink a glass of milk. Now there's lactose in my system. The lactose actually has a binding site on the repressor protein. And then that repressor protein, once it binds the lactose, changes its shape. So instead of looking kind of like a ball glove with little fingers sticking on, it doesn't look like that anymore. And now it can't bind to the operator, and the polymerase can bind and just read the whole way through. So in a sense, the operator is our on-off switch. It is what's going to allow the operon to be expressed or not. And it has to do with whether or not what we're trying to deal with is present or not. All right, that's how the LAC operon works. This is considered an inducible operon, inducing transcription, because we can, when the inducer, the lactose, is present, it inactivates the repressor and turns on the transcription. So in an inducible operon, typically the genes are off, and we have to turn them on. So it's not something we need all the time. It's something we only need when we need it. So we're going to keep it off until that moment arises when we need to turn it on, and then we'll turn it on. There are also other kinds of operons called repressible operons that are basically the opposite. These are in charge of making gene products always. They are needed pretty much all the time. And so you would only turn them off if a given situation arises. So we're going to look at one called the tryptophan operon. All right, now in the tryptophan operon, does anyone remember what tryptophan is? Oh, I just gave you the answer. It's an amino acid. All right, so it's one of our 20 amino acids. And it's something the cell constantly needs. And so the cell has to make it. All right, especially if tryptophan's not available to it from its diet, right, from its nutritional intake. So it has to make it. And there are 5 proteins that are involved in making it. All right, so normally, let me go here. So normally what's got to happen is, here are the enzymes necessary to make tryptophan. In a normal situation, the cell is busy making tryptophan. So this is always in the on position, and this repressor is in the inactive position. But imagine if I drink a glass of milk or I eat turkey, right? So you probably associate turkey with tryptophan. And now the E. coli in my system are just bathed in tryptophan. Would you still want to make tryptophan if there's gobs of tryptophan available to you? No, right? I'm not going to make brownies if my neighbor brings me over a plate of brownies, right? I'm going to eat the ones my neighbor brought me. So in that case, when there's gobs of tryptophan around, we're going to turn off this operon until that tryptophan is used up, right? So what happens is when tryptophan comes around, because of my diet, the tryptophan binds to the repressor, and in this case, when it binds, it activates the repressor, and now that stops transcription. So they're just the antagonists of each other. In the inducible operon, the operon is typically in the off position, and we turn it on when we need it. So that means we're going to inactivate the repressor. In the repressible operon, the operon's on the on position, and we're only going to turn the operator on, the repressor protein on, when we need it. All right? So in the inducible one, the lactose makes the repressor inactive. In the repressible one, the tryptophan makes the repressor active. All right? This is a very efficient way of doing gene regulation because you get what you need in the moment that you need it based on the environment that you're in. It's a very reliable source of getting your information. All right. Any questions on operons? Because that's pretty much all I had to say about operons. Okay, so just make sure you know the two different kinds, inducible versus repressible, and what the promoter's for, and where the repressor protein binds and when. All right, so that was prokaryotes. We're back to eukaryotes, and I just want to talk a little bit more about the complexity of gene expression in eukaryotes. So keep in mind that in many eukaryotes like us that are multicellular, all of the cells in my body have the same genetic makeup. However, I have all these different kinds of cells. I have skin cells and neurons and blood cells and liver cells, right, that all do these very different things. And so therefore, I have to very tightly control what's expressed where and when. All right, so we can regulate gene expression in a whole variety of ways in a eukaryote that allows for that complexity to be very elaborate. All right, so if we look at this, and we're going to kind of break this down, first of all, we have to get some sort of signal. So there has to be something that tells the cell whether we need what we need or not. Okay, so that is part of the regulatory pathway. Is there something telling the cell what you need? All right, so that's one way we can regulate it. If you take that signal away, then the cell's never going to respond in this way. All right, so the next thing we have to look at is the chromatin, right? The change in the structure of the chromatin. We've already talked about when it's really, really tightly wound up into heterochromatin, we don't get gene expression. When we relax it and it becomes the euchromatin, we get lots of gene expression. So what are some ways we change that architecture? What do we add to the histones to make the chromatin get more tight? Anybody?
Doesn't adding a methyl group
Exactly.
make it...
Methylating. That's right. Methylating makes it close up and turn off gene expression. And then how do we open it up? What's one of the ways we open it up?
Something with an A. It's a group with an A.
Right. It's on the screen right here. Histone acetylation, right? And so those are just two. There are actually more than just these two, but these are the two main two that control this dynamic nature of the chromatin. And so this has nothing to do with DNA sequence. It has nothing to do with transcription factors, nothing to do with RNA polymerase. This is just availability. Is the gene itself available or not, right? So we can control gene expression through this. Then we can think about the gene expression at the actual level of the DNA. So let's say we got the signal, we changed our architecture to be euchromatin, the gene is now available. Right now we have to make sure that transcription happens properly. So you need all of the correct transcription factors, you need the proper activator protein at the enhancer, everything has to be, that symphony of things have to be there, the DNA has to bend around before we can get transcription. So there's a lot of control just at that level itself. Once we get the transcription part through and we've made the transcript, we could still decide against what we wanted to do, because we are not going to do anything until we process that RNA. So there's a little bit of control at the RNA processing point as well. If for some reason the cell realizes we didn't really need to do that, we can stop the process right here and stop transcription. Stop the processing and stop translation. So let's just review what are the 3 things that have to happen for RNA processing. What's the first thing that has to happen? I'll wait. I want you guys to be thinking about it. Okay, we have to add a 5' cap, the poly A tail, and then we have to splice it, and that involves the spliceosome, right? That involves energy again. And then, of course, we have to, then it's mature, and we have to transport it out the nuclear pore out into the cytoplasm. Once we get out in the cytoplasm, we can kind of make our decision, is it time to translate or not? It may be that the cell says, nope, we got this far, but we don't really need you, and immediately we'll degrade the mRNA. It could be that we make the decision to go forward, and the ribosome binds to the RNA, it reads through the sequence, it makes our peptide, and then it degrades it, right? Because we don't want this, the length of time, that this RNA hangs out in the cytoplasm will dictate how much protein we make. If this mRNA is only there for a minute, we might not make very many proteins. If we let that RNA hang out in the cytoplasm for 10 minutes, we could probably make millions and millions of proteins. So that's one of the ways we can control it. And we didn't talk too much about this. I think maybe it's coming up. But how do we degrade these mRNAs? Well, that has to do with those little microRNAs that bind to the mRNA that are considered ribozymes that chop it up. They're like kung fu choppers of the RNA to break it down. And then these little pieces will go to the lysosome and get further broken down into nucleotides, which then will get recycled back to the cell for reusing. All right, now we have the protein. Are we done? No, we're still not even to where it's fully expressed yet. So we have to do protein processing, right? We may have to modify the protein. Well, we definitely have to fold it. We may have to modify it. We may have to cut a piece off to activate it. So there's all of those things. And again, however long that protein remains active in the cytoplasm will dictate how much activity happens. If we don't really want it to make a lot of activity, we do a little bit and then immediately we degrade, or we might keep it around a little bit longer and make sure that it's getting its job done. In addition to just folding and modifying, we have to make sure we get it to its proper destination. So is it the secretory pathway? Is it a cytoplasmic pathway? What does that little signal peptide at the beginning tell the cell what to do with this protein, right? And these are all steps along the process that are important for us to consider when we think about, we don't really get protein expression until we get that protein folded and to its final destination and activated. And if we never get there because we stop somewhere along this process, then that whole gene expression pathway hasn't been successful, right? But maybe that was purposeful. All right, so let's think about some examples that we've learned about. All right, so we talked about cyclin-dependent kinases and we talked about cyclins. Remember that cyclin-dependent kinases are pretty much present in the cell all the time, and the kinases are made and then degraded, and then made and then degraded. So at what level do you think the kinase gene expression is controlled? Is it going to be likely controlled maybe at the transcription part of it, where we have the right transcription factors bound and we have it in euchromatin so that we have it constantly on? Or do you think we're controlling it post-translationally, after we've translated it? Any thoughts? That's right. So likely the cyclin-dependent kinase is going to be controlled at the transcriptional level. The cyclin, when do you think the cyclin is going to be controlled? Whether the protein is doing its thing. I mean, of course, we have to have transcriptional control to get it turned on, but what's really in control of whether its activity is important or not? Do you think it's pre-translational or post-translational?
Post-translational?
That's right. We're going to do that post-translationally because we want it to be there for a minute. We need to make the protein, but then after it's done its job, we need to get rid of it so that it's not activating the kinase again. Okay? So those are just some examples of pre- and post-translational modifications. All right, so we talked already a little bit about this, so this is just a reminder that how we go from heterochromatin, which is down here, to a more euchromatin structure, which is up here, and that has to do with modifying those histone tails. So the more methyl groups, the more heterochromatin, the more acetyl groups, the more euchromatin that you have. All right, and so these are enzymes, right? So a HAT is a histone acetyltransferase. So a HAT is going to move the acetyl groups onto the histone tails, whereas a DNMT is a methyltransferase. So they are going to be the ones that add the methyl groups to the histones. And then HDAC, that's a histone deacetylase. So we're going to take it away. All right, so this is just a reminder. Histone acetylation is typically involved with loosening chromatin structure. Methylation is involved with condensing the chromatin structure. If you find oftentimes phosphate groups as well. If a phosphate group is next to a methyl group, that tends to relax the chromatin. So I told you it's more of a real symphony of everything having to be properly placed. But in general, the two I want you to remember are acetylation and methylation. Okay, so you can see here open, look at all those nice little acetyl groups. Here's methylated, you can see all the methyl groups, and that's heterochromatin. So how, what phase or what step in that whole process do histones get fully activated? Post-transcriptionally? When? Post-translationally? So when do you think these modifications happen? You are quiet today. Well, they happen post-translationally. So after we've already made the protein, folded it, transferred it to where it needs to go, then later at the very end, we can modify it in different ways to activate it to do different things. So this idea of controlling gene expression via this histone tail modifications and the changing of the chromatin architecture is something referred to as epigenetics. So it kind of means on top of genetics. It's not just about whether the gene sequence is correct or not. It actually has to do with whether the chromatin structure is proper. And there's a whole field of epigenetic biology that people are starting to understand. This is why just because maybe you don't have a mutation in the breast cancer gene doesn't mean you necessarily wouldn't get breast cancer where that gene's involved, because it could be that it's not the gene itself that has the mutation, but how the chromatin architecture around that gene changes to lead to the cancer. Okay, I think I'm going to skip through that. I think we already talked about that. We talked about our control element. So don't forget, we have proteins that bind to the promoter. We have proteins that bind to the enhancers. And so those are important proteins that need to be present. And I think that was all the farther I wanted to go. Oh, I just wanted to show this picture. Don't forget about that whole bending of the DNA and how important that is. This is just an interesting way to compare. So here are two different genes. Here's the albumin gene, which is expressed in our liver cells. And here's the crystalline gene, which is expressed in our island cells. Liver cells and island cells both have these genes, right? Because every cell has all the same genes. So in the liver cell, you're going to have active euchromatin and bending of the DNA and the proper transcription factors to get the albumin gene on. However, for the crystalline gene, you might have a repressor sitting on a silencer, or you might have euchromatin in that, or I'm sorry, heterochromatin in that area. Whereas it's vice versa in the lens cell, where albumin is off, but the crystalline gene is on. So this is the idea of differential gene expression. Okay, so this is one other thing I want to talk about. We talked about this earlier this semester, and how because we don't have operons in eukaryotes, you can imagine it might get a little bit chaotic to make sure that all the genes of a particular pathway are all expressed at the same time to get an event done. Well, the way it works is that we have these little areas within the nucleus where the proper activator proteins and transcription factors and everything will kind of come and be present, and then the genes for those that are needed all kind of loop out of their additional chromosome and share those same activator proteins so that they're all getting turned on at the same time. So it's kind of like the idea of an operon, because they're sharing the same machinery and kind of doing it all at once, but they're not all on the same chromosome. All right, so we talked about that already. Okay, this is the only other thing I wanted to mention, is we talked about mRNA degradation. And so, like I said, there are little small RNAs. They're called microRNAs or siRNAs, and I'm blocking what the S stands for, that will recognize RNAs that are out there, block them from getting to the translation or to the ribosome, and then target them for destruction. Okay, so any questions about any of that? I know I kind of skipped some of that stuff at the end, but I hit the highlights that I made the test over. Any questions for any of that? No? Well, I will upload this right away. I promise to do it right away for today's lecture. So if you want to watch it one more time before you take the test will open in about an hour, and then you have 24 hours